today's webinar, how United Therapeutics leverages metabolomics to get closer to the phenotype, increasing confidence in clinical trial decisions, presented by Metabolon. I'm Michael Shashua, and I'll be moderating this webinar. Our speakers today are Brian R. Keppler, Director, Discovery and Translational Sciences at Metabolon, and Derek Solum, Associate Director of Product Development, United Therapeutics. Let's begin. Brian, please go ahead.
Thank you. It's great to be here. I'd like to thank the organizers and also Derek for joining me today in this webinar. Basically, in this talk, I wanted to help convey the message on how metabolomics can facilitate pharmaceutical companies in decision-making in drug development. And first off, what I wanted to do is give a primer on what metabolomics is. So what are metabolites? Metabolites are small molecules, compounds that are within our bodies. Most of us know these as glucose, cholesterol, amino acids, but really there's thousands more. They can be tissue type, organ type, flowing through our circulatory biofluids or within our different tissues. And they basically mediate all these different physiological reactions within our body, and whether it's in a healthy state or a disease state. Now, the beauty of metabolites themselves is that they're really the end results of not only biologic interactions, but also environmental interactions. So genomics, or DNA, really gives a good idea of what could be a risk factor. But the question remains, is that gene or, let's say, mutation penetrant, right? Is it transcribed into RNA? Is it translated into protein? And is it then having a phenotypic effect? By looking at metabolites and metabolomics, which are closer to the phenotype, you can get a much more clear picture of a disease state or a health outcome. In addition, metabolomics can provide insight into things like the environment. Does somebody exercise? Do they live in a polluted environment? Do they smoke? Do they have a good diet? As well as factors such as the microbiome. And given the quick uptake in microbiome research in the field today and how gut dysbiosis can be linked to different diseases, metabolomics can really shed a lot of light into the interactions between drugs in the microbiome and also those different disease states. Now, Metabolon, specifically, how we go about performing our global untargeted biochemical profiling is really based on LC-MS technology. So regardless of sample type, whether you're looking at blood or a tissue, the biochemicals are extracted. And again, we're not looking at protein or RNA or DNA here. We're looking at just those small molecules, things between about 50 to 1,500 Daltons. And by running that organic extract across our platform, which is 4 different methods of ultra-high performance liquid chromatography in tandem with mass spec, we can reduce that data down, eliminate the noise, and do a proprietary library matching from a library of over 5,000 metabolites to precisely, with tier 1 identification, determine which metabolites are in a sample. And based on study design and appropriate statistics, you can then start to make comparisons on the effect of a drug or the effect of disease versus healthy, for example. Now, in Metabolon's platform, the visibility that you can get is very comprehensive, and you'll get a good understanding of that when Derek gives his talk about the data he received from some of the studies that United Therapeutics has done. As an example, when we're talking about amino acids, we're not just talking about 20 metabolites, the 20 proteinogenic amino acids. There's about 400 metabolites within amino acid catabolism that is in Metabolon's library. So you're looking at not just the parental amino acids, but how are they metabolized? How are they used? And that type of depth can really provide knowledge about what's happening within a system. If you go along the different super pathways that are involved in this comprehensive view, you can look at carbohydrate metabolism and energetics and central carbon metabolism, cofactors and vitamins, nucleotide metabolism, lipid metabolism, as well as some novel metabolites, and as I mentioned before, some metabolites that are microbiome related. And the tricky part about biochemistry is they are all interconnected, all of these pathways, one perturbation of one pathway can have distal effects in another, right? If you affect or block, or if a disease causes let's say, an inhibition of a certain enzyme, you're going to have a backup of substrates. Those substrates are going to want to go somewhere. They might go to an adjacent pathway. So by understanding all these super pathways and the interconnectedness, you can really start to dive deep and elucidate the true effects and those effects that are causing the phenotype that is visualized in, let's say, a disease or a drug effect. So in terms of pharma drug development, the applicability of metabolomics is really very diverse, can address many of the pressing needs that many of our clients in not just pharmaceutical development, but applied markets and biotech work face. It can be from wellness to disease. It can be looking at biomarkers of efficacy or safety, prediction of disease, simple understanding of biology, understanding the microbiome, the genome, patient stratification as well, identifying responders and non-responders, and then, of course, optimizing drug development, which we'll focus on a little bit more on this talk. And when we're talking about optimizing drug development, it's the idea of finding or identifying the 5R framework for those of you who are familiar, identifying the right targets, the right tissue, the right safety, the right participants, that patient stratification that we talked about. So if you take a look at here, really what we do at Metabolon in terms of trying to drive and facilitate decision-making for our clients in pharma and drug development research, metabolomics is really applicable across the continuum of the drug development process, from discovery and target selection all the way through clinical. In discovery, you can look at things like target identification, right? You can also apply this with reduction of genomic data. You can have lead candidate selection where you're identifying predictive biomarkers or doing some early tox identification, maybe finding in vitro off-target effects. In the preclinical space, again, looking at biomarkers, maybe homology and translatability across mammalian models. We've also done a lot of work in metabolite-directed compound assessment in preclinical models. And then moving translationally into clinical, trying to find a drug's mechanism of action, a biomarker of response, an assay development validation, resistance mechanisms, and then, further downstream, patient stratification, trying to really enrich a population with those patients that are going to respond to a therapy. Maybe even trying to do repurposing or combination potential of a drug. And really what doing such a broad, global, comprehensive look at metabolomics does is it changes the established repertoire from a simple drop in the bucket, if you will, of just looking at the drug, its metabolites, and maybe some expected pharmacokinetic or pharmacodynamic data into more of a complete view, right? Seeing all the puzzle pieces together, right? Imagine if you have a large puzzle and you're missing some of the key pieces and you have an idea of maybe what's going on, but if you don't see all the interconnectedness, then you might be missing something. And I like this analogy too with the ripple effect and drops in a bucket, because when you have more drops, as you see in the bottom picture here, those ripples start to affect each other. And that's really what happens in biology and physiology and the etiology of a disease. And that's the type of elucidation and interrogation that metabolomics can provide, really trying to take that data and turn it into knowledge and insight and really dissect all the interconnectedness of a disease. A little bit about Metabolon's library. As I mentioned, it's a library of about 5,000 named metabolites. These are all small molecule entities in many different diverse superclasses and subclasses, as I mentioned. In all the different projects and different species that Metabolon has analyzed with our various clients over the years, one thing to note is there's many different sample types that can be analyzed: plasma, serum, urine, different tissue, feces, saliva. And in doing a study and really employing a study design that is constructed to meet a certain objective by doing different tissues, and Derek will touch on this a little bit in his talk. Let's say, looking at circulatory biofluid and a tissue, you can get a more complete picture of what's going on. And I just want to leave you, before I pass it over to Derek, with one example of the power of metabolomics in marrying with another key omics technology in genomics. And this is an example that is showing how different SNP variants can be identified. So extracting the value from human genetics. And the challenge here was really trying to unearth the next PCSK9, right? These rare genetic sequences that are associated with diseases and treatment responses, right? Sequencing is a default tool. It's a great tool for finding targets, but the question is what target or what gene should be focused on? And in this study, sequencing and metabolomics were used together. The two omics were married, and the test case was high blood pressure, right? So can metabolomics identify the important genes faster? And what was identified in this study by looking at a large population was that the loss of function variant, or the mutation in the SLCO1B1, was leading to increases in a metabolite, a dicarboxylic fatty acid called hexadecane dioate. And you can see in the middle section of the slide here, the box plot, you can see with the number of loss of function alleles, hexadecane dioate increases significantly in the association for SLCO1B1. And what they found was that by marrying these two omics technologies and the data, that there was an association, a very strong association between increased blood pressure and risk of heart failure, and that this hexadecane dioate was increased as the disease severity increased. And hexadecane dioate is actually a substrate for different oat transporters, right? And basically, the findings from this study were able to really validate a new paradigm for targets and unlocking human health and disease, identification of new targets, and providing avenues into gene function and the understanding of disease etiology by integrating these two omics technologies into a deeply phenotyped population. And I just wanted to give you this one example of how metabolomics can be used, again, as a primer for some things that Derek's going to talk about in the next part of the talk. And with that, I'll pass it over to you, Derek.
Excellent. Thanks, Brian. I hope everybody can hear me. I just want to say thanks for inviting me to chat about some of the work that United Therapeutics has been doing in the metabolomics space and the precision medicine space. This is something that we spent a lot of energy on recently, so it's great to be able to speak in a forum like this as well. I will kind of carry forward here, and I wanted to kind of give a little insight into United Therapeutics, because I don't know how many people on this call are familiar with the company, but basically United Therapeutics was started by Dr. Martine Rothblatt, who is a lawyer and businesswoman initially, and a visionary and really an entrepreneur. And it's kind of her interest in satellite communications that ultimately led to her development of Sirius satellite radio, which I'm sure we're all very familiar with. Martine's daughter was actually diagnosed with the disease, pulmonary arterial hypertension. And just like any parent, she basically took her kind of business acumen and entrepreneurial spirit to sort of to go after that disease. And that's really what she has done and what has led to the development of United Therapeutics. So along the way, in order to sort of find cures for this disease, pulmonary arterial hypertension, United Therapeutics licensed a molecule called treprostinil, and treprostinil has now been through clinical development and is one of the really one of the initial and great drugs used to treat the disease, and there's a number of different formulations with that. In 1999, United Therapeutics went public, and this allowed the company to really continue to grow. But over the last couple of decades or so, United Therapeutics has grown, and there's a number of different subsidiaries, primarily located across the East Coast. I'm located in RTP. The headquarters is in Silver Spring, Maryland. But at the end of the day, UT, as we refer to it internally, is really founded in science. And I think that one indication of that is if you actually look at the image in the kind of top right, you can see that the walkway between a couple of the buildings and manufacturing facilities is the DNA double helix. So I think that provides insight into really how UT keeps its sort of core goal on the focus on being scientific at heart. And along the way, UT has not also lost its focus, and that is we primarily continue to do research in pulmonary arterial hypertension, and that is the focus of most of the work, and most of the work we've done in metabolomics. And so in order to kind of understand, I would like to just give you a little primer about that disease. And so pulmonary arterial hypertension is primarily idiopathic. The causes are not totally known, but it has a lot of different factors that contribute to it. It's a multifactorial disease, really, and a number of genetic implications have been identified, but they're like most diseases. There is no one gene, one disease aspect to this. There are mutations in what's called the bone morphogenic protein receptor, and that causes a lot of people who have those mutations to ultimately have the disease, but there are people who also have the mutations who don't get the disease. So it's complicated. And this is like a lot of diseases, but there are a number of risk factors that are ultimately involved, and those are presented here on the slide, some of them anyway. Whether it's collagen vascular disease or heart disease, there's also drug and toxin related. Fen has been implicated in the disease as well as some infections as well, including HIV, have identified as risk factors that can ultimately lead to PAH. The pathophysiology is complicated, as I mentioned, and it's also, like I said, multifactorial. There's many aspects to this, and really. The long and short of it is that you get changes to the pulmonary vasculature. So this is the arteries within the lungs. And in a normal individual, the endothelium, which are the cells that sort of line these arteries, they basically facilitate the normal biological activities, blood flow, inflammation, cell growth, and they do this by secreting a number of factors, and those are included like nitric oxide and prostacyclins, other molecules like endothelins. When this goes awry, when this signaling kind of gets out of balance, that is when you start to get what we refer to kind of as endothelial dysfunction. You start getting remodeling of the endothelial vasculature, and that's kind of shown in the cartoon here. And the challenge with this disease is that it's chronic and it's progressive. It continues to get worse, and ultimately with this vascular remodeling, what you get is a narrowing of these vessels. And so the blood vessels within the lungs ultimately the arteries get smaller and tighter, and it reduces the blood flow, and this ultimately continues and leads to increased pressures in the right side of the heart, and ultimately that causes a muscular remodeling and an atrophy within the heart, and in many cases the consequence is heart failure. And so it's a complicated and it's a chronic progressive disease. But at the end of the day, there's a handful of signaling pathways that have been implicated and is the primary focus of most treatment. And that, as I noted, is the nitric oxide, the prostacyclin, and the endothelin pathways. Ultimately, treatment hits those three pathways, which is the nitric oxide, the endothelin, and the prostacyclin pathways. And it's those three pathways that kind of have been the focus of getting most of the treatments to people who have pulmonary arterial hypertension. the challenge really becomes that even though there are a number of treatments available, and since last 25 years or so, 14 different therapies have been approved by the FDA in order to treat people, but just like many diseases, it's not so simple that there's one treatment that works for any particular individual. This cartoon basically just shows you how this disease is multifactorial, and that there's a lot of different signaling pathways, whether that also includes genetic abnormalities or other biochemical or other biological modifications. But ultimately, as I mentioned, it leads to a chronic progressive disease where you get this restriction within the pulmonary vasculature. A number of diseases or a number of treatments are available, as I was mentioning, but the challenge really becomes that just like almost all drugs, most all pharmacotherapeutics, is that they don't work for everybody. And typically drug development uses large clinical studies through many phases of drug development. And when those studies prove successful, that ultimately leads to drug approvals, but not everybody responds. And in fact, of the 10 top grossing drugs, of all the people that do benefit from them, usually between anywhere from 3 to 24 people who were treated with them actually don't receive the benefit that was intended. So it's kind of, like I say, this is a one-size-fits-all approach from therapies. And so the goal is to really, from a precision medicine standpoint, is to how can we tailor our therapies so that the right people are getting the right treatments. And that really is the concept of precision medicine, and that is not so much that we are going to try to develop a therapy that works specifically for an individual. That's more a concept of individualized medicine or personalized medicine. But precision medicine is a concept that we want to sort of stratify individuals based on some characteristic, whether that may be drug response, drug tolerability, or some sort of prognostic factor. But ultimately, the goal being that we want to stratify individuals in order to make sure that they get the therapies that are going to work for them. And so as I mentioned, there's a number of ways to stratify people. And, some drugs people can't take because they don't tolerate them very well, or in others, it's just that the drug response doesn't work for them, and maybe that they have some other predisposing factors that limits that. But in our process of precision medicine, really, the concept looks kind of like this, where we take a broad population, as shown kind of up on the top row, and some of these individuals are going to be considered, say, responders. And then from this broad population, we collect biomarkers, and those biomarkers can be any number of things, and I'll show in a moment, but they can be blood molecules or metabolites, as Brian has mentioned. They can be some sort of specific polymorphism within the DNA. They can also be other phenotypic characteristics or clinical variables as well. So some sort of biomarker is collected, and that's one of the goals of our precision medicine program, is to collect a broad panel of biomarkers in order to help us stratify. So then, using the evidence from these biomarkers, we stratify individuals and essentially assign people to a category of where we think that they will be, that they can benefit from the therapy or not. And then ultimately, you have to test that. So you do these in clinical studies in order to validate this. And this is the concept really of how precision medicine will work. And so, as I was saying, biomarkers can be any number of things. We use various biomarkers in our clinical studies right now. They might be clinical lab results, other phenotypic characteristics. There's a variety of things that can be used, but the biomarkers at the end of the day measure something, and that's some sort of pharmacological response, or some sort of measure of disease progression, for instance, or any sort of change based on a therapeutic intervention. Biomarkers, as I mentioned, are pretty broad. They can be some sort of pharmacodynamic biomarker. We also use a number of surrogates. In many cases, it's difficult to have a direct analysis of a tissue, for instance, so we use some sort of surrogate endpoint. There's also prognostic or predictive biomarkers, which can sort of help provide insight into disease progression or whether or not an individual actually is susceptible to the condition. So we use these biomarkers kind of as a surrogate in order to evaluate people. And so at UT, our precision medicine process is really goes into a number of different phases. And I kind of alluded to it in a couple of slides ago, where really the first thing that we do is to collect samples, right? So we're going to collect and analyze pharmacogenomic and biomarker samples, such as metabolites, from the people who are in our clinical studies. So these are people who are provided the information, and then they consent to have these biomarker samples collected, and so we collect them. The next is basically to compare the molecular characteristics of people who respond and those who don't respond. That really gives us an insight into who our responders are. And then the key is to correlate these biomarkers or these sort of metabolomic or pharmacogenomic signature with the clinical data, because, you know, at the end of the day, our clinical studies have clinical endpoints that are defined and discussed along with the regulatory bodies to determine whether or not the therapy or the treatment is effective. And so after that is done and you've validated and compared to the clinical response, the goal is to validate that predictive signature, and then it ultimately create some sort of companion diagnostic so that you can indicate whether or not prior to treatment whether or not somebody will be effective at receiving the particular therapy. And this has been going on for a little while in the oncology space, but within many other disease states, this is still relatively new, and so it's gaining hold, I would say, and it's becoming much more widely used and accepted. And the approach we've taken really is kind of a pretty broad one, and that is that we use kind of a pan-omic or a multi-omic approach in order to evaluate individuals in the studies. And Brian sort of gave a little bit of a hint to show that there's really this biological cascade, the central dogma, if you will, where DNA is transcribed into RNA, and RNA is translated into protein, and then through various biological processes, you get metabolites. And so we've taken an approach to where we're kind of looking at all of these, where we look at DNA through genomics and RNA through transcriptomics, proteins through proteomics, and lastly, metabolites through metabolomics. And the reason we want to take kind of a broad swath here is that, as I mentioned early, is that we have a multifactorial disease, and there are some sort of genetic dispositions to this, but it's still relatively unknown. And most of the people who are diagnosed with PAH, the reason is not known. And so we are trying to look at this, and ultimately metabolomics is one of those pieces to the puzzle. So we're trying to fill in a big puzzle, and every piece is essential in order to get to our results. And this kind of gets at the idea that by using this pan-omic approach or this multi-omic approach, the plan is to basically create some sort of multi-omic fingerprint or some sort of molecular signature, and that this signature will allow pharmaceutical companies in order to identify individuals who are going to be responders or people who will tolerate the therapies or people who will help stop the disease from progressing. And this is really the approach that we're taking, is that we take a number of different pieces and kind of from that Venn diagram really how do these various omic pieces, how do these various biomarkers come together in order to provide insight into a number of different things, not just disease state, but really response to therapy. So as I alluded to before, there are a handful of, couple of decades, there's really been a lot of changes to the ability of people to sequence DNA. And I'm sure there's plenty of people on this call who were doing Sanger sequencing back in the day. We've come a long way since the human genome was completed in 2003. And not only has the techniques become faster, but it's also become more efficient and much more cost effective, which allows us to do this. And you can see across the top, at least within our disease state, there's a handful of genes that have been involved, but ultimately we still don't have kind of the that smoking gun, if you will. So we continue to pursue the various approaches in order to identify what really is the cause for the disease. And this kind of leads us to adding metabolomics to our kind of portfolio in our multi-omic process. And the reason being is that, as Brian alluded to, metabolites are really a downstream consequence of what is actually coded for in the DNA. So you can get variants within the DNA, and ultimately those lead to protein variants and changes in metabolism. And so it's kind of a link, and not only does it give us insight into the genetic makeup, but it also gives us the potential to analyze samples relatively quickly and relatively easily without having to do broad genetic screens or genetic sequencing as well. And really, I think the beauty of metabolomics is that it provides kind of an insight into the biochemical state and the normal physiology. And this happens sort of, throughout disease progression, whether you can compare normal to those who are at various stages of the disease, that this sort of readout is possible. And that's one of the nice things, is because we do get biological changes as the disease progresses. And it's not unique to pulmonary hypertension. There's many diseases that follow a very similar pathway. The other advantage really is that it can provide a surrogate marker of the disease state and then help us monitor the progression and responses to therapy. Another advantage really, and I'll allude to that in the next slide, is that it gives us a broad signature, really. And this is one of the reasons we've added metabolomics, is that normally within our clinical studies we collect a variety of clinical laboratory tests, that is, blood samples that look at liver function and a number of other factors. But that's pretty small, relatively speaking, when you think about all of the different metabolites and all the different ways that you can look at and analyze an individual's sort of biological activity. And the other real advantage is that it can be assessed in a number of different tissues, right? So Brian suggested that, they can do assessments in tissues, but one advantage for us is that you can have tissues that can be collected via minimally invasive procedures. And this especially is helpful within the clinical trial space because it allows for easy sample collection, easy storage, and then we can also ship and analyze pretty quickly. And the other thing we can do is we can analyze across different types of tissues, right? We can look at the plasma or serum or urine, or, if we wanted to, we haven't done fecal matter to look at the microbiome, but those are all possibilities, and they're relatively easy to collect those samples, and they provide a pretty robust readout of what's happening. But kind of the long and short of it is that adding metabolomics has really helped us so that we can, think about translating the science from the initial stages to through the clinical development. And it's also fruitful in the way that you can identify biomarkers and then help to study the disease and disease mechanisms and provide insight into that as well. So as Brian showed this slide before, and this is one of the things that really kind of got our interest, really, is this broad panel of metabolites and these different metabolic pathways that are covered within the global platform. And I'll show you that we've done some studies now, and some of these aspects are very well related to the diseases that we study. As I alluded to at the beginning, PAH is a multifactorial disease. It involves inflammation and vascular remodeling, and there's oxidative dysfunction, and so there's a number of ways that we can look at the disease progress through metabolomics. And so this broad pathway kind of is helpful from that regard. And when we started looking and developing our kind of pan-omic approach, we wanted to go at this kind of from a really agnostic way, is that we wanted to not have any focus and let previously determined genes or any outcomes limit what we would look at. And so that was why we went to look at this kind of broad pathway with the global platform. And so within PAH, so I mentioned this is still relatively new, doing metabolomics. There has been some work which is been very helpful in addition to ours. But the main details, as I was mentioning, is that being a complicated disease that impacts a number of different pathways, metabolomics has been able to show that there are changes in the glucose and energy metabolism, as well as fatty acid oxidation and glutamine metabolism. The other thing is that there's been some reports that shows that dysregulated circulating metabolites have also been associated with negative outcomes in PAH. There's also been some non-clinical research in this space, and there's some recent studies that have shown that you can actually, in the non-clinical space, in the rodent model, you can actually see that post-treatment you can see a variation in the metabolomic signature. So this is very interesting as well. But within the clinical space, there's evidence that shows that the individuals who have PAH are different from a metabolomic standpoint from those who do not have it, and that those metabolites can then be used in a prognostic way, and that those individuals can, you can identify them, and ultimately these people have an increased risk of mortality. So it's very useful from a biomarker standpoint in order to understand the disease progression. And the nice thing is that the metabolic state really can provide a close representation of their overall health. And so there's, all of these things are kind of leading to increased interest from the clinical development side of things as to using this type of biomarker as a measure of responsiveness. And so we've done a number of studies, as Brian had alluded to, and we started this a few years ago now. And some of the initial results we've published, and actually we presented this with the Pulmonary Vascular Research Institute meeting earlier this year. Seems like an eternity ago now, but ultimately we've sort of been collecting biomarkers and collecting samples from subjects who consented in our phase 3 program, at least for this presentation. And then sort of the goal to be, how can we correlate treatment response with the changes in metabolites? And from a couple of different phase 3 studies, so these are global phase 3 studies, the answer to that actually was yes, and that we can sort of look at from a metabolite standpoint, and then correlating that to the clinical response, we can actually see that there's kind of a different fingerprint, if you will, from those who were treatment responders from those who are placebo responders. Obviously, the placebo responders are not really what we're looking for. We want to get that signature from people who respond to therapy. And what is actually quite interesting, so there's a couple of the numbers here are shown in these two Venn diagrams, where in one study we had 69 specific metabolites that were statistically significantly different. In the other study, there was 58. And what's quite interesting is that there's very little overlap between the treatment responders and the placebo responders. But similar to what was identified with sort of the normal versus the disease state is that these metabolites really kind of fall into categories that are not a surprise, and that is that they are related to hypoxia, glycemic control, lipid metabolism. There's some microbial architecture activity as well as host redox status. So we've got a number of categories where we've seen the metabolites change, and this is kind of presented here, which shows the actual metabolites that were shown to be different, both from a perspective of baseline, which is the panel on the left, versus the post-treatment, because they're both really of value. But we do want to be able to get to where this baseline activity is informative, because we want to be able to look at an individual prior to treatment and determine whether or not that individual is going to be a responder. So you can see that there are a number of different metabolites that were identified, and it's easy enough to bucket these. In our initial work, we were worried that there are going to be thousands, but that did not turn out to be the case, which is great. But you can see from the list that most of these are identified. Brian alluded to the fact that within their platform, they've identified most of the metabolites, and there's some that are as yet unidentified or unconfirmed. But we have a primarily identified metabolites and some that haven't been. But this kind of gives us good insight into that signature for being able to identify a responder. And those metabolites are kind of bucketed here. And so in this heat map, what you see really is in the sort of all of the columns that are under the orange, those are varying levels of response. So in our studies, there's a couple of ways we sort of measure response. And so if we look at just varying ways of those response, you can see that those each of the bars represents a change in some sort of metabolite, with red a metabolite that's significantly up, and green a metabolite that's significantly down. And you can see that in the green bar across the top, those columns, those are all of our treatment responders, and the orange bars are our placebo responders. And so you can see even in this heat map that there's a difference, and it looks like that category of treatment responders has a generally speaking a different kind of pattern than it does in the placebo responders. So it gives us insight into really who those responders are and how we can identify them. So that's been very useful. Kind of a summary from the metabolomic studies we've done is that we've done the global platform and looked at hundreds of people in our studies, and from those initial findings, we've detected about 1,400 distinct biochemicals within the plasma, and similar amounts. I didn't show any of the other tissues like the urine samples that we've collected, but ultimately we were able to, from those samples, all of those numbers, able to identify a signature that really helps us to pinpoint who is a responder and who is not a responder. And ultimately those categories of metabolites that were shown to be changed are pretty similar and well within what we would anticipate, and those buckets kind of being within the hypoxia and glycemic control, because keeping in mind that this is, it's a pulmonary disease, so any sort of change in oxygen utilization could be manifested here. So we have a number of different categories that are involved, and these are all similar to what is related to how the pathophysiology of the disease is. And so that's been very useful to have metabolites and metabolomics kind of as part of the puzzle that we're trying to put together, really. And so kind of in the long and short of it is really this is how we're going about the precision medicine, and that includes the broad pan-omic approach that we're taking, and that is individuals from our studies. We ultimately are collecting samples. The samples need to be prepared and analyzed, and then there's an informatics piece as well, which I didn't really go into, but obviously this is, it's complicated in order to identify the various differences between the samples, and then ultimately identifying a fingerprint and getting that fingerprint to be able to kind of evolve and validate it, and the later phase really is and the goal for us is to get to where we can develop companion diagnostics to sort of evaluate individuals prior to treatment. So that's my last slide. I think we do have a few minutes for questions. So Michael, I don't know if you want to, how you guys want to do this. You open it up.
We do, Derek. I'll open it up now to the Q&A, and there's still time to submit questions using the Q&A tab on the left side of your screen. We have lots of great questions already, and we will try to get to as many as possible. So the first one here for you, Derek, is actually got a few parts, so if I may go one at a time here. What hurdles in United Therapeutics drug development and clinical trials did Metabolomics help you overcome?
So that's a good question. So we face all sorts of hurdles all the time, and I would say that the one piece that the Metabolomics is adding is helping us to identify responders. This is still a bit of a work in progress as well, but I think that that's one real advantage, and I think that moving this precision medicine piece forward, I think that one of the advantages and a hurdle really is being able to conduct clinical studies efficiently. I often think that, the studies 10 years from now are going to look very different from studies now, because right now we often conduct very large studies, and they take a long time, and so it takes a lot of resources, both from the developers as well as the subjects, and so we keep that in mind, and so that's relatively important. So I think that helping us design better trials is really one of the pieces that will be valuable.
Okay, so what has Metabolomics provided that other data streams could not?
So again, good question. So I think that the value that Metabolomics provides is more of a sort of dynamic readout. Genomics is a different piece where the DNA doesn't change in an individual over time, whereas the metabolomics do. And so with the disease progression, and as I was sort of alluding to at the beginning of the presentation, is that the disease we primarily study is a chronic progressive disease, so it can provide insight from the beginning of the disease to the later stages. Okay.
And then how did recommendations and paths forward derive from Metabolomics data influenced decision making?
So I think this kind of goes back to the inputs about how it will inform future clinical studies. We like I said, we ultimately use the data from one study, and that informs the data for how we're going to design the next study, whether that be the type of endpoint or the type of samples that we collect or analyze or anything along that line. So I think that every piece of data like this is very valuable as far as informing how you're going to do your drug development or how you're going to do your clinical development program.
All right. Our next question here is, most data presented from United Therapeutics is work done in phase 3. So is United Therapeutics considering overlaying metabolomics earlier in the drug development process?
The short answer is yes. I mean, the initial studies that we've done are phase 3 studies. We do have other early clinical studies ongoing, and the goal really is to gain insight from subjects who are in the studies. So primarily after you get into phase 2, where you start actually seeing patients rather than healthy volunteers. So this is really where I see the future.
All right. There is a follow-up here on that. Is where do you see metabolomics fitting into United Therapeutics R&D and clinical workflow going forward?
Yeah, so I think it will continue to expand our, multi-omic exploration. So I think that we're still just on the cusp of really understanding how the multi-omic approach can be used. So I think that it will continue to be a piece of that puzzle at the end of the day, informing us of how to recruit and stratify the studies, and then also the readout as well.
Another one. So was there an instance where metabolomics data was the first time you received data towards specific phenotypic indication?
No. We've sort of added metabolomics on to the clinical development program as it's, sort of been underway. So we've had, obviously clinical development goes through, it's many years in the process and even includes a lot of non-clinical pieces as well. So we usually have a chunk of data that is giving insight. So it's at least for us right now, it has not been we haven't had any insight up to this point, but that may change.
Actually, so we have one here for Brian. Even if a drug is not influencing its primary or secondary endpoints, can metabolomics be informative on paths forward?
Yeah, so that's a great question. And I guess to answer that, I can give you some examples where we worked with clients in the pharmaceutical realm, not just in working with drugs that are currently in a certain phase of clinical trials, like Derek provided his example for, but for drugs that have quote unquote failed, or drugs that have come across hurdles in FDA acceptance, and the clients have used that as a reason to perform an exercise and use metabolomics to identify why. What could they have done differently? What biomarkers maybe could they have identified? What different tissue types might they have utilized instead? Of course, plasma and serum are very common. Derek mentioned that they did urine in United Therapeutics as well, although he didn't focus on it in his data. But metabolomics is applicable to cell culture. You can do cell pellets. You can do spent media or supernatant. You can analyze liver tissue, kidney tissue, the full gamut. Anything you can harvest biologically from any living thing, you can perform metabolomics on. So, incorporating that extra visibility, that extra data stream, as I mentioned, can help bring those that maybe blurry picture into more focus, make things a little bit more granular.
Okay, thank you for that. The next question is for the placebo responders. Did these people really present improved symptoms? Did they have relief from the diseases? And also, what were the biomarkers that were consistent with disease relief comparing to the drug subset? I'm not sure if I can follow all that, but maybe you can unpack it.
So the question is really asking how the placebo responders differed, really, I guess, if I'm kind of following the thought stream. Well, I mean, we all know that there's placebo responders regardless of the sort of study, and our studies are no different. The real goal is to be able to identify a treatment effect from those who receive the active drug versus those who receive the placebo. And so then the question of, is there really a difference in the type of biomarkers? I would say, the reality of it is that you need to look at a kind of fingerprint. It's not going to be a particular metabolite or a particular gene that's going to be unique, but rather than how is the package different, and that's really the goal. So from that perspective, we see a difference between the placebo responders and the treatment responders. So I don't know if I answered that question properly.
Well, I think so. I think
I got all the
can
pieces.
follow up if there's more on that. So how will metabolomics change how clinical studies are conducted?
Yeah, so I kind of alluded to this a little bit. And for us, I think the goal is to stratify the studies so that you get the right subjects in the study. And so I think that kind of moving forward, we'll be able to use not just metabolomic data, but multi-omic data as well to have smaller studies. So we're enrolling kind of the right people. And then I think that they'll be also informative from an endpoint standpoint. So looking at how changes are, how the treatment is affecting disease. And so I think that those will be changes that we definitely see. And precision medicine in general, I think, will be playing a big role in this in the years to come.
How can precision medicine, including metabolomics, be used to monitor a patient's disease progression?
Yeah, I think I kind of covered this as well earlier, but I think that the nice thing is that the disease we study is a progressive disease, and you're constantly having biological changes, that involve various pathways from inflammation and vascular changes. And so I think that being able to have some sort of readout on that is valuable.
Okay. What's the long-term stability of metabolomic samples?
Yeah, I can take that
for
one.
Me, maybe. Yeah, good. I wasn't sure who that's directed to.
yeah, I can take that one, Derek. So metabolites are fairly stable, but just like any biological sample, you want to collect them and store them appropriately, right? We have recommended collection and storage protocols. Things stored at -80 are stable for, we're not talking about days or weeks. We've done samples that have been stored over years, which is of course very important when you're talking about working with clinical samples that have been collected over long periods of time. So I'm happy to have discussions with people on that. But collected and stored at -80 and shipped on dry ice, and really consistency is really the most important thing. The metabolites, the reactions which metabolize those are going to be pretty stable for years, the year time frame. So, and that's regardless of matrix type.
Okay, this next question will be our final one. We had a lot of great questions today, and we could not get to all of them, but we'll try our best to get back to everyone who submitted personally after the webinar. And the question is, have you been able to verify the metabolites using quantitative assays from the same samples? Is this a necessary aspect of the validation process?
For me, the answer is no. Our samples are collected kind of under a specific consent, and so we kind of, we can't. They're collected with the knowledge that they're going to be used for a particular assays, and then usually we don't, store them and then go back and do various quantitative assays. So we have not done that. Whether it's a necessary process of required for validation, I guess, that's a question. I don't know that it is, but ultimately, it may be important to have some sort of validation piece to it.
Right.
Just to add to that, the tool that, the main global untargeted biochemical profiling that was used, that Derek used and United Therapeutics used for Metabolon is, it's a great hypothesis generating approach to give you insight into changes in disease or changes with treatment. Metabolon also can provide fully targeted quantitative assays for which, given the client's desire, can be performed for validation purposes as well.
Great. Thank you, gentlemen, and thank you all for attending this Fierce Biotech webinar and submitting so many great questions. I'd like to thank our speakers for participating and Metabolon for presenting today's webinar. This webinar has been recorded. Thank you again for joining us, and we look forward to seeing you at future events. you.